we explain clone. So what's clone? Clone is individuals or cells that are generally identical. Next, what's cloning? Cloning is the process of creating a genetic copy of an organism. Let's see history of clone. In 1885, Hans Adolf Roech experimented using chia gene. Two cell embryos separated each cell and then grew into a complete cell gene. He succeeded the first artificial cloning of animals. Then in 1996, Jan Weymouth experimented with the use of sheep. He transferred the nucleus from an adult sheep's udder cell into an enucleated egg, and then its egg grew into a sheep, which called Joey. He achieved the first mammal cloning made from somatic cells. After that, cloning of mice, cows, and rhesus monkey succeeded. Researchers had tried to use clone of other organisms.
Next, let's explain how to make a clone. There are two ways to make a clone. The first method is to use embryonic cells in the early stage of fertilization. In the method of using embryonic cells, first after fertilization, one cell is separated from the divided cell that has 16 to 32 cells. Next, the cell and the unthird fertilized egg are electrically stimulated to cause cell fusion and induce cell division by culture, and then return to uterus. After a while, clones are made. The gene of embryo cells generated from one fertilized egg are all the same, and individuals produced using these cells are all clones having the same gene. However, at present, embryos with advanced cell division are not suitable for clonal production, so the number of clones that can be produced is limited. On the other hand, in the method of using adult somatic cells, following this process, the composition of the gene is almost identical to the gene of the original somatic cells. Also, since there is no limit to the number of somatic cells that can be used, it is theoretically possible to produce an unlimited number of clones. That's all.
Cloning is considered to contribute to various fields. Let me explain some examples. First, livestock industry. By using cloning, we can raise a large amount of good quality livestock at low cost. As a result, we may be able to eat more delicious meat. Second, regenerative medicine. To apply cloning to ES cells technology for making organs attracts more attention now. In organ transplantation, by using our own cells, rejection doesn't occur, and patients don't have to wait donors. So, this technology saves many lives. These two examples are under development. When these are put into practice, that would be a great advance for humans.
Next, we talk about ethics problems. As you know them by some news or books or movies, in 21st century, we humans can make clone animals, including humans. So now we face difficulty not how we can create them, but how we should treat them. Is it okay to make clone animals? And in particular, what about humans? Improving science research. When making clones, we can predict their appearances and type of expression in a certain degree. But there is a risk of regarding people coming to the world as means or tools. Moreover, because of their existence, the way to be born, our paradigm of birth slightly change. To summarize, making cloned animals, especially humans, will cause the problem of dignity. It seems there's no direct effects, but we have to forbid them.